Thank you everyone for coming. My name is Yee Man from Anti-Genetics. Tonight I'm going to talk about cure cancer through next generation sequencing. Let me start with a self-introduction. I'm a bioinformatician with 10 years of experience. Bioinformatician is a professional who writes software to analyze DNA sequences. I was also part of the Human Genome Project. And you can find my name on the chromosome papers published in Nature. I personally have friends and family who contracted cancer, so I always wonder if there's anything I can help them. Coincidentally, cancer is caused by changes at the DNA level, so indeed I do have a role to play in the fight against cancer. So this is nowadays how a typical cancer patient, when they visit an oncologist, their tumor sample will be sent to a lab, and then a few genetic tests will be conducted. A test report will be returned to the oncologist, and the oncologist will prescribe targeted therapy drugs accordingly. Instead of just testing for a few genes, we propose to test all the 22,000 genes in the human genome in one go. In this picture, we are the computer guy who received the 60 gigabase sequencing data from the lab and returned a report of 22,000 genes to the oncologist. Why do we want to test so many genes? Physiologically speaking, cancer is caused by gene changes that lead to uncontrollable cell growth. At least 500 genes are identified to drive cancer growth, and new cancer genes are being discovered as we speak. By testing all the genes, we can increase the chance of a patient being eligible for targeted therapy. So let's take a look at this stomach cancer subtype pie chart. Currently, there is only one genetic test, HER2 amplification test indicated for stomach cancer. So at most, only 22% of the patients can be eligible for targeted therapy. By testing all the genes, we can increase the percentage to 42%. Only KRAS or PIK3CA type stomach cancer currently do not have eligible targeted therapy. We envision that as more and more targeted therapy drugs are being developed, more and more patients will be covered, and eventually majority of them can be eligible for targeted therapy. Before I talk about a cure for cancer, let me tell you more about targeted therapy. Targeted therapy is designed to target gene changes that only occur in tumor. Therefore, compared to chemotherapy, they are more effective and have fewer side effects. In fact, many patients can live a normal life while under targeted therapy treatment. However, as some of you might know, targeted therapy eventually becomes ineffective and people succumb to the disease. So, cancer really is a tough nut to crack. Is there any way we can go about it? After reading conceptual papers about cancer fighting strategies, I come up with a cure for cancer. By cure, I mean turning it into a livable chronic disease like AIDS. First, tumor DNA is inherently unstable. So over time, tumor evolves and acquires DNA changes that make it resistant to targeted therapy. So we propose periodic tumor resequencing to adjust targeted therapy treatment over time. Second, research indicates that targeted therapy resistance usually caused by DNA changes in multiple genes. So a rational combination targeted therapy that involves multiple drugs targeting multiple genes should be able to overcome it. And indeed this is the case according to some small-scale research. By doing periodic resequencing and combination targeted therapy, we can maximize the chance of a patient finding effective treatment. As long as they are eligible targeted therapy drugs, patients can live a normal life while waiting for the next breakthrough in targeted therapy and achieve the cure status I outlined earlier. So enough about the technical stuff, let me talk about the business side. In 2010, there are 26,000 new cancer cases in Hong Kong. Since our service is quite expensive and targeted therapy is also quite expensive as well, we anticipate that only the top earning 4.5% can afford it. So we estimate that there are about 1,000 annual customers. But our market can also grow by itself thanks to the following developments. After we gain traction in Hong Kong, we have plans to expand to other Asian markets. And our market will also grow by itself thanks to the following developments. DNA sequencing cost is falling exponentially. More and more targeted therapy drugs are being approved. Targeted therapy drug prices are also falling steadily, and the awareness of cancer genetic testing is growing. All these contribute very favorably to our business. As to our current status, we have developed a ready-to-use software pipeline to generate a report that recommends targeted therapy drugs based on tumor DNA changes, based on DNA changes. So if unfortunately your family or friends contracted cancer, you can talk to me after the talk to learn more about our service. We are ready to help right now. We are fundraising right now, and I'm looking for oncologists as a business partner, so if you know any oncologists, please let me know. We are not the first in the business. 2 months ago, Foundation Medicine IPO to a market cap of 900 million. As you can see, the revenue grows tripled every year. The main difference between us and them is they sequence 236 genes, but we sequence all the 22,000 genes. I think their success indicates this can be a viable business. To conclude the talk, I would like to reiterate that comprehensive tumor genetic testing is key to cure cancer. The macro trend is favorable to our business development, and the success of Foundation Medicine suggests we have a proven business model. That's the end of my talk. I would like to take a chance to thank my parents for the understanding of me not having a real job. I also owe a lot to the free software and database developed by the research committee, and finally to CoCoon for this nice co-working environment and giving me this chance to talk to you. Thank you.